Oh, hey guys. I'm Jacob Gray. I'm a rising junior at Michigan State University, studying under Dr. Ganz in the Zebrafish Lab. And I'm a first year Choose Development Fellow. So this is the main room where we do most of our regular lab work, like labeling PCR tubes and writing PCRs and lysis and our thermocycler, as well as setting up stuff for gels and whatnot. And today is one of the days where I'll be running a gel. So I ran a PCR the prior day, put the my samples, and then go into our gel room to then rerun that gel, to then run that gel for the PCR spec yesterday. To do that, I have to go to a separate room we have called our gel room. As you can see here, it's a nice big room. So while I'm loading these PCRs, I can tell you a little bit more about myself. I'm a rising junior, as I said earlier, at Michigan State University. I'm on the bowling club team for Michigan State. I am also in a separate program called MSU SRP to foster graduate studies. And I'm also interested in going into cephalopodal research, specifically developmental biology and behavior for my graduate and postdoc research. So with our gels all loaded, you can see it's got those green bars is where the wells are. It's gonna be like 2 rows of bars. Now all we have to do is throw the lid onto the gel tray, run a current through it, and then we'll come back later to check up on it to see if it ran all through. So while I'm getting this lid placed onto this tray, I'm having trouble here. As you can see, we can transition back into the main room to then go ahead and look at our fish room. So the fish room has some special rules where we can't take our regular shoes into the fish room. So what we have to do is we have to use our Crocs to go into these fish rooms. And we got Lysol and whatnot, and you can't go into the fish room with the Crocs, with your shoes, and then vice versa, can't do the Crocs outside of the fish room. So in the fish room, we have 2 racks of fish, as you can see to the left, and they have all this desk space where we use to breed fish, as you can see here, to do fin clips and whatnot as well. And as you can see here, one of our my colleagues, has already got some fish set up. It's like the wild type and cross right here. And then right here is a lot of the desk space that I usually work at to do my fin lysis and whatnot. And then as you can see in these orange bobbles are our team we feed to our nursery fish. Transitioning to the racks we look at, you got 6 rows of these tanks filled with our zebrafish, and then we'll go into what my actual fish look like. And these are the carriers that I have to identify and then cross to get our baby fish that I looked at later. So now we go into the gel room again to see the DNA has properly gone through our gel, and that's good application. So then we're going to transition upstairs to our other lab and use our gel doc to then look at the actual amplification of our DNA. And as you can see, the gel doc is on here. All right, so what what's going on here is get ready to put my gel into our gel doc so I can see the results. So once we have the gel in there, after we position it so that it can get a clean read, and then we run it through our program here, and then once it's run, we get like a picture of where the bands are located to see the gene expression. Once you get the pictures, we print it out, but we don't dispose of the gel because it's a special kind of waste. We have to put it in a separate waste container. So we'll throw that over here under there. And then once we throw it away, we wipe down the actual tray, and we take our container back downstairs to our gel room. Alright guys, that was a little tour of my lab. As you can see, I've changed venues. I'm at home now, but working in the lab's a lot of fun. I can't wait to do more research in the lab. Also can't wait, looking forward doing cephalopod research with squid, octopi, cuttlefish, or any of those organisms because they're really cool. And yeah, I'm really excited to keep working with yous, and I'm grateful to be here. Thank you for listening. I'm Jacob again, and I'll see you later.